the ATG gene function. 2016 Nobel laureate, Professor Yoshinori Osumi at Tokyo University. We had some dinner 3 years ago at Tokyo. He clearly explained the model animals' importances in Japan Korean society. This is one of example. This is ATG5 translating mice. Using translating mice, maybe autophagy can be acquired to the community. It can be a good clue for making a new therapeutic agent for new disease and different types of disease. So this is one of the example because Osumi sensei, Dr. Professor Osumi's main tool is just yeast. There is a really strong, powerful tool for making a basic science result. However, sometimes for human application, we strongly need more humanized animal model, including mice. This is very good example. Also, okay. As you know that already, PLOS Biology Journal is very important and a high qualified journal. Some specific and a very interesting paper was published in PLOS Biology. That's economics of reproducibility in preclinical study. NIH wanted to know producible or reproducible data from NIH-funded animal experiment. The result is very interesting because 0.5% is reproducible and 0.5% is irreproducible, even though that project has been supported by NIH funding. However, 50 and 50, just the reproducibility. So what's going on? The major reason is, just poor quality of biological samples and a reagent, mainly simply cell lines. Yeah, just to skip. Poor quality of cell lines. Well, sometimes we believe this is A cell line, however, B. Sometimes we believe this is a B cell line, male cell lines, however, sometimes females. So the reproducibility is deeply dependent on the qualification of a sample, qualification of resources cell. So NIH wanted to boost up the higher qualification of a bioresource, especially including cell lines and organoid up to animal models. This was, introduced 3 years ago in Nature Index because Korean government spent a lot of money, especially for R&D budget. So the fastest way to reach a high quality of science by governmental side is one of the answers is just they focus on biological resource programs. Maybe there was a study 10 years ago. This is one of the answers. So this is one example. And 4 years ago, Nature made a special volume for Korean scientists booming up in Nature Index. Our center was introduced in one of good examples how Korean government wanted to spread out the higher quality of bio infrastructure in Korea. So today I wanted to share 3 issues. One is my center KMPC activity, and second is like Cellpedia in France, we expanded the other types of animal models from xenophage to non-human primate. That is second one. And third one is, just during pandemic crisis, we well organized, just a preclinical study. Before I introduce this one, I just wanted to briefly introduce our story in Asian area. In Asia, EMNA is an Asian Mutant Mouse Resource Association. Currently, I'm a president of EMNA meeting. And in every year, so we have some workshop for postdoc or some graduate student. That is, weekend in Japan, MIR's Mark in China, and KMPC Korea, we have some meeting. Also, we invited several speakers from outside, including Centroid here, maybe just too small. You can see your face here. And Colin Macaulay. There is, really good window for communicate with the other communities. I wanted to show up this one, maybe center. This is me. This is Dr. Shiroishi, current president of KMPC. We met in 1995, almost 30 years ago. During rest 30 years ago, we published some data and we have some workshop. We have organized, some scientific program. That's a very important because sometimes, just global science should be dependent on local small community. So this is one example. And this year, Obata sensei, Obata is, just a former president of RIKEN BRC, retired, and I joined that official retirement, ceremony issues. So this is very small, but very cozy story. Also, we wanted to expand our issues. Surely we joined several IMPC meetings, including Finland and Australia. Also, just a center-to-center meeting. For example, we have some meeting with Czech CCCP. Also, we have some meeting, ICS. IMPC is a very big umbrella. So we have a very clear and distinct final goal. However, also at the same time, we wanted to share our experience and our knowledge very detailedly for advancing our science. So that kind of small group meeting is very good. Also, we already invited Steve, and we have some meeting 2012. This is our first time to communicate with the IMPC members. And 2021, including and a very long flight from South Africa to Korea. Anyway, we have some meeting. And then we expanded that IMPC meeting with a Nature publication groups and a Nature Metabolism editor-in-chief. We invited him and we co-organized very special topics. Okay, my center, very simple generation mouse models and mouse phenotyping. And we can share that data by portal. Also, my center is very briefly, in a year, we have some $50 million just for 10 years guaranteed program. And several research topic areas, including metabolism and exercise, and sensory, including vision, hearing, and olfaction is our major goal. Also, we manage the germ-free mouse system, also in vivo molecular imaging. And this is one of the issues to communicate with the young generations. This is just we call the mouse card. And we make some illustration for each mouse, including genotypes, including Bayes phenotype data. So we can circulate that across the community and across the classmate. And this is our most representative metabolic phenotyping platform. We focus on energy balance itself. However, as you know, already, just the behavior and exercise issues. Also, immunity is one of the key elements to solve something issues even in metabolic phenotypes. So integrated, platform for mouse phenotyping. That means, we already published several data. One is STK3 knockout and a TM4S5 mice. That means, we generate mouse models to our friends. Also, we support their mouse phenotyping in our center. This is a very good platform for advancing our colleagues in science, also for ourselves. This is one of the example. Vision. Very easy for human to detect vision. Can you see or not? Just a simple question. However, we can ask to the mouse, can you see this or not? This is alphabet A. We can ask that. So we have to use just a behavioral test combined with vision. So there is one of example. So this kind of data was formulated in just mouse portal, like this. Okay, let's move to national bio infrastructure in Korea. This was also introduced in 3 years ago in Nature Index. KMPC is just a mouse center, mouse research center. At the same time, KMPC is act as a centralized biobank in biodiversity program in Korea. We started almost maybe since 2009, just the initial stage. And from 2011 to 2020, we called, phase 1 period, almost 10 years just to Korean government support to expand the number of biobank. At the same time, we don't know which resource is important. We don't know how we can share that valuable resource maintained and used by our colleagues. Just collect that. However, we suddenly realize this kind of effort is very not so much effective. So we wanted to change that. So 274 biobank, including potato, including just small vegetables, including small burgers, and we make 14 clusters. Nowadays, I'm a headquarter of model animal cluster at the same time. At the same time, head of mouse center. We have 14 bioresources, including cell lines and including brain tissues, including microbiome or natural resource chemical library, even in some cases, fishery organisms and marine organisms. So simply, I mean, animal model is very important because sometimes we need a very low levels but high throughput techniques. Even I'm a mouse specialty. Sometimes I need some genophus, I need some Drosophila. However, it is not easy even for me to use just different types of animal model. So resource and education and analysis is needed for spread out this kind of infrastructure. So this is one of the example. My center is a KMPC, centralized animal center, and a mouse and a rat and a genophus and a zebrafish and some sea elegans. This kind of animal models, I mean, cluster was organized by my center. And just to simply, I mean, just to very user-friendly, I mean, collection and sharing for their own research purpose. Because I mean, we are mouse provider. Sometimes I mean, users, I mean, needs cannot be emphasized because I mean, we are just we don't have enough energy for more than just collection. However, without users, our resource is useless. So even though the size is very smaller, however, very useful mouse models, I mean, meet the new needs from our colleagues is much more important point. So we already have some non-mutations almost across the human whole genome. However, in some case, they need just knock-in or they need just a human genetic variation, even same genes. That kind of new needs should be met ourselves, by ourselves. And finally, I mean, we just to introduce a COVID-19 crisis. This was also introduced in Nature Index. Very simple. In the early stage of 2020, at the time, we don't have mouse models for spontaneous infection of COVID-19. So we have to develop that. And then we circulated that, we provided that mouse models to our researchers. But as you know, as already, I mean, COVID-19 research should be done within animal biosafety level 3. So that means, I mean, very low capacity to provide a preclinical study. So there are several variations and barriers. So we make some alliance among ABSS laboratories. So we supplied, I mean, standardized, I mean, K18 humanized transgenic mice to them. And they infected their mouse. And then these all mice could be analyzed in 1 or 2 pathology centers, including molecular imaging, including omics. So meet the new needs from researchers. We validate, I mean, different types of promoter, humanized transgenic mice. As you know as that, in mouse models, I mean, they cannot be survivable. Just to 90% were dead in 1 week after infection. This is a very good example for testing vaccination because we can just calculate number of survival, number of death after vaccination. It's very easy. However, it's known as that just less than 3% of COVID-19 patients cannot be survivable. They are very severely, heavily damaged in lung. So to research their real severe pathogenesis in lung, we need a very, I mean, different issues because, I mean, in mouse models, I mean, survival rate is too low, almost to 10%. This is good for vaccination. However, this is not good for long COVID. This is not good for just long resting, I mean, severe lung damage. So we developed, the lung-specific promoter with the humanized transgenic mice. One is SFTPB. There's upper levels of promoter, and CCSP, there is lower levels of promoter. And K18 and KEG is, ubiquitous promoter. However, unbelievably, the result is very different because K-humanized transgenic mice survival rate is very low. However, compared with that, K-humanized transgenic mice, survival rate is a little bit higher compared with K18 because K18 showed just brain damage levels. However, KEG mice, they didn't show the brain damage brain damage area. That means depending on needs of corona researchers, maybe we can validate the even simple mouse models with different promoter. Also, this is just to process, but it can be standardized, and just protocol is standardized. This is very beautiful picture. Maybe you can see easily a real virus particle in mouse lung. This is, conducted by Professor Kyung Seong, Yonsei University. And also, this is a very good example. Upper panel is just to low magnification, high magnification. In viral infection, you can easily found a very severe immune cell invasion here. However, YG1, this is a new treatment, and YG2 is another treatment, and YG3 means just a combination cocktail of YG1 and YG2. You can found just local inflammation here. However, after treatment YG1 and YG2, unbelievably, there is no severe immune reactions there. So this is one of a successful case in our preclinical platform. So we evaluate more than 100 candidate drugs and new candidate drugs and just vaccination. We tested 53 and we got some 13 positive result from them. And this kind of all of data can be stored, has been stored in LIMS, Laboratory Information Managing System. That means in ABSS3, in 6 different ABSS3 lab, they store that all of data in portal. So that was managed by LIMS, and that can be visualized by this portal. So you can see easily here, just 3,896 mice. Yeah, their raw data was stored there, and just without exact name of therapeutic agent, you can easily access all of data, just body weight and just Wuhan or Omicrons or just infection route. You can validate all of data there. We did some omics analysis. The red color means already published Berkeley, single-cell RNA-seq, also ribosomal profiling. That is closely related with the RNA methylation changes by SARS-CoV-2. Also, just red means just under submission or just under processing. I briefly introduced several impact of data from single cells. You can see easily this is a damaged lung. This is spleen. This is very interesting because day 1 and day 2, you can easily find the white pulp. White pulp means just lymphocyte population's reaction. However, after 5 and 6 days, white purple's morphology has been destroyed. This is very unique findings from mice. However, in human, we couldn't find that because we cannot take spleen. Yeah, that's the issues. But this is closely related with how they can work within a body, destroyed by COVID-19 infection. So simply, this is a single cell data. Oh, very interestingly, just monocyte and PDC is unregulated in early stage. However, CDC is unregulated not only in early and late stage. However, macrophage is unregulated in late stage. That means, up and down regulation according to each cell population can be very precisely managed. Interestingly, this is just myeloid population. This is day 0. You can easily find a very big portion. This is macrophagy. However, macrophagy pipa gamma. That means tissue resident macrophagy. However, after infection, the population is seriously decreased. Instead of that, green fraction was increased. There is a monocyte Ry6C2. That means circulation monocyte that can invade into the lung after infection. And then later stage, you can see easily just a sky color. That means a monocyte. There is a macrophage SPP1. That means, pink color is also macrophagy. However, they are tissue resident macrophagy. However, new population marked by sky blue color, there is, change from monocyte to macrophagy. So that means, most invaded macrophagy population is not tissue resident macrophagy. They are come from just blood circulation. This is one of the good evidence for making what cell we should make a target. Also, this is I just want to skip this one. And nowadays, Wuhan is the original strain. However, Omicron is, just popular. So we already experienced there is a bit of different after infection of Omicron and Wuhan. So we wanted to find some changes in single cell levels, very clear. This is myeloid population. This is Wuhan, and this is Omicron. This cell population is quite different. Red means upregulation. However, this is not upregulated in Omicron. Which cell? This is TNF alpha and NF kappa positive myeloid cell population's regulation is unregulated in Wuhan, however, not unregulated in Omicron. We can easily conclude just to total context of immune reaction may be lowered in Omicron virus compared to, with Wuhan's. Also, this is, same situation as lymphoid. And NK cell population's reaction is very strong in Wuhan, however, not strong in Omicron. Okay, totally what's going on between Wuhan and Omicron. This is one of the simple data. This is Wuhan, 0, 1, 2, 5. There is no 7, 10, 14, because they are dead after 1 week later. However, Omicron, they cannot dead within a 1 week. They can be survival up to 2 weeks. So you can see this, you can see this population here, 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 here. So this population maybe changed and never returned in Wuhan population. However, in Omicron, that areas changed, and after 14 days, they changed to normalize, very similar to day one. That means just based on trajectory issues, especially DATP. DATP is damage associated transient progenitor cells. That is very one of the key elements to reading to lung damage and lung regeneration. That reaction is quite different between Omicron and Wuhan. Also nowadays, long COVID is very important because COVID patient population is much larger than the obese population in the world. So this is one of example. This is normal lung. This is impacted lungs. Maybe you can easily see very atrophic brain regions after SARS-CoV-2 infection. Also, we wanted to found very interesting correlations between survival rate and immune reaction and brain damage, because the very advantage point to us, we infected more than 3,000 mouse models in our center. That means very big cohort, even though they are not human. So we summarized that data, and this was published in the American Journal of Pathology. So this is KEGA human ACE2. There is no brain reaction, no infected in brain. However, K18, they impacted in brain area. So very different. So K18's survival rate is severely lower than K, and there is closely related with the immune reaction and a specific cell population, CD40 positive cells. Also, just I wanted to briefly summarize my short talks. We develop animal models COVID-19, and this is very organized mouse genetics, including mouse generation and mouse pathology and infection and immunological phenotyping. That is very helpful for organize this kind of meeting. Okay, thank you for your interesting and attention. Thank you.
Thank you very much, JK. Very impressive presentation. Any questions here?
Kent, yeah. Kent first, yeah.
Thank you, JK. Beautiful talk. You talked a lot about the transgenic, like your CAG, you know, but you know there are some deficiencies with the transgenics with multiple copies and so on, and where they're expressed. But you didn't say anything about your knock-in mouse or your dual knock-in ACE2 TMPRSS2. Did you want to say something about that too?
Oh, okay. There are 2 points you already mentioned. One is, copy number variation. Some people told us just the copy number is a key issue, so reading to this one, regardless of promoter. However, still, negative data opposite to that result. And secondly, just to knock in. That means, human ACE2 transgenic mice with mouse ACE2 promoter. However, even though we make a mouse ACE2 promoter and human ACE2 transgenic mice, the viral distribution is not too much different with the others. So it depends on the what science we want. Yeah, but just we wanted to make a very simple and a reproducible animal model for COVID-19 and long COVID. So even the basic issue is a little bit different, just we want to make a very reproducible survival rate and reproducible lung damage. So a little, just viewpoint is a little different. Yeah.
Thank you for your presentation. I liked it, but I would like to add something. We also should talk maybe a little bit about politics. And the politics it's not about I would like to mention that very recently on the 11th of April, so just 2 weeks ago, 3 weeks ago, the European Union, and I was involved in that in Brussels, has the first signed a contract between South Korea and the Horizon Europe program. 95 million is included. And I think that program could bring also collaboration between our organization here and your organization.
Yeah.
And we should use that. I was noticed that if we send any kind of application to do something, joint workshop, exchanging students, exchanging PhD students, we could use the money now. So really we can help you with that one. So this is an important step. And first, and then the second part was, I like and we're going to discuss a little bit more later around your brain part, of course. I was interested to see how COVID-19 is infiltrating into the brain and if there are specific brain areas. And then my final one is a question to you. How is your institute integrated in Korea with KBSI, the Korean Brain Science Institute? Can you tell us a little bit about that?
Okay. Korea is a very small society, so everybody know each other. Especially, the director of KBSI, Korean Brain Science Institute, and Pan Gil-sung is one of my long friends. So we can share our resource, and also we can share our project for making a good result too. But just I'm not a specialist in brain science, but just I have a very new and simple questions on brain issues in COVID-19. I always ask to them, and some part they are interested in, some part they are not too much interested in it, depending on that. I wanted to learn from clinical side because animal model is similar, have quite different in detail issues. So I wanted to know detailed progress and a detailed event in human, and then I wanted to apply my system to meet the questions. That's my answer, sir. And our first one is, this is very new emerging issues. Maybe Korea, we want to have a very new issues with the EU. That means we can apply horizontal program, so we can be one of the members. Also, that means Korean government, President Yoon has a great enthusiasm to expand our research with the global society, including USA. So we have some another program with USA. Also, we have another program for EU. Anyway, it's a really good chance for us to making some small step and a big step with the European countries. So please, mail to me, and I want to make good relationships. Okay, thank you. Yeah,
Great. Thank you. Any more questions? No one. So thank you very much.
Okay, thank you for your question.